That's what I was fearing you were going to say.
So can I just cancel this update? And perhaps cancel. Or I will.
Yeah, it's just now I want to disconnect because I want to go through them,
Yeah,
but I'll
please.
disconnect and can I disconnect and reconnect?
Because it is in live streaming. Hopefully it should be fine.
Oh, I see. I'm in Zoom.
Yeah. No. Should be fine. Okay.
Oh, okay. So then I can go to my.
You can
Oh
just
no.
take a seat there and then do that. But copy that one. That's fine.
That's all right. I can go through it.
You can take a seat and bring it.
No,
to-
I'm.
Okay, fine. Yeah.
Sort of.
Okay. Please, here.
Oh. Shall I share?
Yeah.
Oh, yeah. Let me just. Sharing.
Okay. Welcome everyone in today's public lecture. So at the beginning, I will request Director TIFR Professor Jairam Chengalur to start the today's event. Professor Chengalur.
Yeah, good afternoon and welcome. I'm delighted to welcome all of you to TIFR. As you probably all know, research at TIFR covers a very large range of areas, everything from the largest scales of the universe to understanding the interaction of subatomic particles, to studying complex physical, chemical, as well as biological systems at all the intermediate scales. And in many of these areas, TIFR is often the only institution in the country where studies in this particular field is actually happening. And so we've always regarded it as part of our mission to try and communicate to the public at large the excitement of doing cutting-edge science. As you probably also know, TIFR has centers all over the country, and all of these centers have their dedicated outreach programs. So, for example, here at Colaba, we have been running for decades now the Chai and Wai series of public talks at the Prithvi Theatre, as well as other locations in the city. And of course, we have a long tradition of hosting public talks here at the Homi Bhabha Auditorium. So I'd encourage all of you to also check out our webpage as well as our social media feeds for the list of upcoming events. Today, we are fortunate to present the latest event in this series, a public talk by an extremely distinguished speaker, Professor Janet Thornton. You'll shortly hear a more formal introduction of the speaker, but I'd like to take this opportunity to thank her for accepting our invitation to visit TIFR and for giving this talk. And in addition to this talk, she'll also be spending time at TIFR, not just here on the Colaba campus, but also at NCBS, and will also spend some time interacting with our faculty. So thank you, Professor Thornton, for making the time for this visit. I'd also like to thank the Sarojini Damodaran Foundation for very generously providing the support for this visit. This kind of philanthropic support is very important, not just in carrying TIFR's mission forward, but also in taking it in directions which are otherwise difficult for us to explore. So with that, I'll stop and I'll hand over to Debashish. Welcome again.
Okay. I will now request Professor Chengalur to felicitate Professor Thornton. Yeah. Jairam. Okay. Now it's time to tell a little bit about Professor Thornton. So it's with great pleasure now I introduce this year's Sarojini Damodaran Eminent Fellow, Professor Janet Thornton, who is the Director Emeritus, the European Molecular Biology Laboratories, European Bioinformatics Institute in Cambridge, UK. This visit of Professor Thornton is supported by the Sarojini Damodaran Fellowship for Academic Visitors of Excellence, funded by Shibulal Philanthropic Initiative and Shibulal family with support from TIFR. Let me tell a little more about Professor Thornton. Professor Thornton graduated in physics. After that, she has done masters in biophysics from King's College, London, and a PhD in biophysics at the National Institute for Medical Research, Mill Hill, London. After doing a postdoctoral research in Oxford, she moved to London before becoming the director in MBL EBI. Professor Thornton is one of the world's leading researchers in the structural bioinformatics, using computational methods to understand protein structure-function relationship. So she works at the interface of biology, physics, chemistry, and computational computational biology, computation. This includes a detailed analysis of enzyme biocatalysis, their mechanism, and the evolution of new functions. She has published more than 500 papers. She was elected to the Royal Society in 1999 and is a foreign associate of the US National Academy of Sciences. In 2012, she became a Dame Commander of the Order of the British Empire for services to bioinformatics. Today, Professor Thornton will be talking about the contributions of bioinformatics over the last 50 years in deciphering structure-function relation of enzymes and their evolution, how it is relevant for health and disease. Without any further delay, I invite Professor Thornton for this talk. Thank you.
Visible. So it's my great pleasure. Oops. To be here or to be in India, which is always both a pleasure and an adventure. And it's also great to visit TIFR here in Mumbai. I've never visited Mumbai before, and it's an amazing city. And here you are so lucky to be on such a gorgeous campus that I'm quite envious of having the sea so close by. But today I decided to speak in particular about enzymes, evolution, and disease. And it is a very personal reflection on some of the things that we've been doing over the last 50 years, which is really when I started doing bioinformatics. And I think you probably all agree that we have been really living through a revolution with the publication of the human genome in the early 2000s. And I think the importance of this really cannot be overestimated in terms of our understanding of biology and understanding how life has evolved. But of course, the challenge is to interpret those genome sequences in terms of their biological function. We know that we as, oh no. Okay, so this is for the people online, I assume. So we as humans have over 20,000 different proteins, not very many considering how complicated we are. And our challenge is really to try and understand how life works with this new knowledge that we've acquired. So could I just ask, can you hear me at the back? Yes, it's all okay. I realise you probably can't see very much of me, but I am here. So I wanted to talk about enzymes. So enzymes are one class of proteins that perform all the chemistry of life. And the reason they're so important, there are many reasons why enzymes are important. The first enzymes were used many thousands of years ago in food production for cheese and bread and alcohol. Enzymes themselves were discovered in about 1833. We had the evolution of the word catalysis that is so important by a Swedish scientist. A Danish scientist then produced Rene, which is used for thickening, and a German scientist coined the word enzyme for the Greek enzymos, which means to leaven the bread, raise the bread. And then Buchner discovered cell-free fermentation, i.e. that enzymes on their own could perform this catalysis. It didn't have to be in the cell. And so there's a long history of enzymes. And looking back again, even in the 1950s, from 1900 to about 1950, enzymes were the reaction performed by the enzymes were defined by these reaction diagrams, where you have what I call the substrate changed into a different product or two different products in this time. And we have the uncovering of the metabolic pathways, which, given how little we knew about enzymes before 1950, is quite amazing that these scientists could develop this complex network. But we also knew that there were many different metabolic pathways. So enzymes, and this is important. So the reason why, in fact, we came to study enzymes in detail for about the past 20 years is because there's more data for enzymes, more structural data and also sequence data than for really any other type of protein. And so if we want to understand the relationship between structure and function and evolution, this was a good place to start. And we know a lot about enzyme reactions because of the work performed by the Enzyme Commission, who assign 4 numbers to each enzyme reaction. So this doesn't depend on the protein sequence, it's purely on the reaction that is catalyzed by the protein. So we have these 4 numbers that define a reaction, and there are 7 major classes. The last translocase is a relatively recent addition. The majority of enzymes are oxidoreductases. They transfer things, or they hydrolase. They're to do with water. So then, about in the 1965, we not only got the first structure of an enzyme, Henneguac lysozyme, and I was fortunate to be in the lab with David Phillips, who you can see here, and Dorothy Hodgkin at Oxford, when they set up after the lysozyme structure had been developed, and also after Dorothy Hodgkin had determined the structure of insulin. And we not only have now structures of many enzymes, but we also have information about their kinetics, binding constants, the Michaelis-Menten equation that describes the balance between product and substrate, and this classification. So from 2000 onwards, we have enzymes galore, if you like. And in fact, this was, if we look in GenBank or for the number of publications in public PMC, Europe PMC, we can see that we have many thousands of publications in each year. We can also see that we have now structures for over 100,000 different enzymes in the protein data bank. And as I said, they're really important proteins because if we look at plants, then about 36% of plants are genomes code for enzymes. For human beings, it's a bit less because we have more control proteins than for example, in E. coli, where 37.7% of the genome codes for enzymes. So we had a goal. So this could be given the name computational enzymology, which is rather a sort of posh name, for trying to model the reactions of these enzymes in the computer. And so our goal was, first of all, to capture, to analyze, to validate, and ultimately to try to predict enzyme mechanisms and to discover how enzyme function in a family of enzymes changes during evolution. Because we know that if you take one enzyme with a given function during evolution, that family of enzymes, the gene will duplicate and you will end up with enzymes with new functions, new acquired functions. So if you want to take a new enzyme, generally a new enzyme will be evolved from one that we've seen before. So we started doing different things about enzymes, as shown here, the different sorts of considerations that you might want to do, including evolution, flexibility, families, reactions, mechanisms, and ligands and kinetics. But before we could start doing anything, we needed to gather the data from the different databases, because although we have a lot of data, in general, it's scattered around in different databases. So we created something called the Mechanism and Catalytic Site Atlas. It started out as a very simple project that was called the Catalytic Site Atlas that merely took an enzyme in the protein data bank, the structure of it, and annotated those residues that were involved in catalysis, defined what we meant by that, and annotated it. And that then developed into a more sophisticated database that include information about the mechanism. So in the MCSA, which is available at the European Bioinformatics Institute, we store reactions, a set of reactants and products. We store information about the enzyme itself, the UniProt entry, etc., the Cath classification, the species. We store the information about metal ions and cofactors that are involved in catalysis. And most importantly, we stored those residues that participate in this catalysis. Now, this seems very trivial. It is very trivial. At the time, though, the only way you could get this was to go and read the papers. And when you have, as we now have, about 1,000 different enzymes, reading 1,000 different papers isn't much fun. And so you need to be able to compute in it and use this information to look at enzymes' active sites. But not only that, we also added, or Antonio Ribeiro added information about the mechanism of the enzyme, how the enzyme actually performs its catalysis. And with all this data, here today we have actually just over 1,000 enzymes in the MCSA, of which about 3/4 have encoded mechanisms. So those are the curly arrow diagrams. I'll come on to those. So we can immediately ask, well, proteins are all composed of the 20 different amino acids. Which are the most common catalytic residues? So we can see here the amino acid type and the residues, what you see immediately, and then we can normalize according to the number of amino acids in proteins as a whole. But we see that histidine, cysteine, aspartic acid, glutamic acid dominate in terms of performing catalysis on a small molecule or on another protein. So we can gather the statistics about which are the important proteins. And you can see, of course, that the first 5 are all either positively or negatively charged, and that's important. And then the later ones are polar. So it's the electrostatic forces in general that drive catalysis. We can also look at what are the detailed functions of the side chains. Now, as you will have heard from my CV, I trained as a physicist and not a chemist. So I always struggle with some of the chemistry. I'm sure maybe there are others in the audience who also struggle with some aspects of chemistry. But fortunately, I've had very good people in the lab who understand the chemistry. And one of them, this was mainly Gemma Holliday's work, defined a set of roles for the side chains. And you will see from this distribution of different functions, and I don't know whether you can read them along the bottom here, it's quite difficult. But the thing that really struck me is that most of these mechanisms are driven by proton acceptors and proton donors. And that is at the basis of green biological catalysis. And we can also, I won't go into this, it's too much detail, but we can also say which amino acids like to do which function, which chemical function. And you can see that the amino acids, if we compare going down, actually perform, some of them are the same, some of them are different. In particular, cysteine can perform a lot of electrophilic interactions and radical interactions that can't be done by other amino acids. So this is useful information about enzyme catalysis. So what I'm going to do now is to tell you a little bit about how enzymes evolve different functions, and we'll look at paradigms of enzyme evolution, studies of the active sites, and some rules of catalysis. So we know for evolution that the way that proteins change over evolutionary time is usually that a gene, shown in yellow here, is duplicated, and then one of the genes has a new function, neofunctionalization, and so then we have 2 genes which are related to each other. They all come from the same original gene, but they perform different functions. So the question is, okay, we know this happens. What sort of processes are involved when we look at different families of enzymes? And if we ignore that for the time being, we can say that for the P450s, what most enzyme families, the way they evolve, is they just act on different substrates. So they evolve to bind different substrates. So the P450s are a family which has exploded. That it's they basically, especially in plants, where at this point when this was written, there are about 1 million total P450 sequences known. This is one family of enzymes. And basically, this family in the P450s constitutes about 1% of the coding genome of plants, because what they do is detoxify, and they're sort of hormones, they perform all sorts of different roles. The P450s act on different things, but only a very small number of these new sequences are defined. If we look at the substrates of this family, what we see is there is a huge variety of substrates. And when I started, I really couldn't believe it, because in one's mind, I had a picture of an active site, a binding site that bound one sort of molecule, and that was what the enzyme did. In fact, during evolution, that binding site evolves. So in this case, for the P450, it can act on very different molecules, from quite small ones to quite large ones here. And so understanding those is really important. So that's the most common paradigm of enzyme evolution. You change your substrate, you evolve the ability to bind new substrates, different, sometimes related, often with a common core. So another way of evolving new functions is actually seen in the pseudoenzymes. So pseudoenzymes aren't enzymes. They've usually lost their catalytic activity, and that usually happens by a mutation in one of the 3 catalytic residues. And one mutation, if you lose one of those critical amino acids, you can no longer perform catalysis. So you effectively lose a function. So then the question is, well, why are these pseudo enzymes retained? And the reason is that they have other functions. They evolve, they can't do the catalysis anymore, but it turns out they can be useful, especially in regulation of all sorts of, so regulation of the enzyme itself. They bind the substrate, but they don't catalyze it. So it's like a buffer that buffers the quantity of a substrate. They regulate localization. They regulate recruitment of different substrates. And most importantly, perhaps they regulate transcription. So these pseudoenzymes, where the enzyme has lost its ability to do catalysis, nevertheless are very useful in biology and have been used and retained for all these different reasons. Now, there's another way that, again, I didn't realize was even possible, but we're working on this family of enzymes, the triterpene synthases. And all of these enzymes, and there are many, the way this works is it's like a pathway, and the triterpene synthase generate different scaffolds, but they start with the same substrate. So these enzymes all bind the same substrate, but the product they produce differs according to the sequence of the triterpene synthase. So you don't change the substrate, you still bind the same substrate, but you generate, you can see here, different products. This is the original substrate. And there's a carbocation cascade, which is complicated. These, as they're called, scaffolds, are then further decorated by other enzymes that in total generate 20,000 odd structurally diverse triterpenes. And these triterpenes are the hormones of the plants, but they're also the defense mechanism, which of course plants can't run away, so they have to chemically defend themselves. And these triterpenes are at the heart of that defense. So another paradigm of enzyme evolution is that you know that enzymes, all proteins, have domain structures. So they, on average, the domains are 200-300 residues long, and you don't just get a bigger and a bigger enzyme. You grow an enzyme, if you like, often by adding different domains. And what you see, and is shown here, is that many proteins evolve by the fusion and swapping of domains. So you have a 2-domain protein, one will be constant, and then you change the other domain, so that you get multiple variants of that. And this actually shows the radical SAM family, which has a very specific function. But all of these little squares show different combinations with that core domain that performs the reaction associated with SAM here, and that's the SAM molecule here. And so you can see there are all sorts of different domains that grow out, and perhaps you have one new domain, then you'll get 2 new domains and sometimes 4 new domains. So you can mix and match your domains to give enzymes that can perform related but different functions. And lastly, of course, if we think about the serine proteinases, so proteinases are very dangerous molecules to have lying around in the cell because they chew up proteins. And the last thing you want is that these serine proteinases or any proteinases go and destroy all the proteins. And so over time, this family has duplicated. There are now many different members of serine proteinases, some of which are, I should say, the serine proteinases are one family. Well, no, actually, there are 2 families. Cetylysin and is very different. But what I want to point out here, this is for polymer specificity. So defining a specific protease that will degrade a specific protein needs some sort of specificity. Some proteases will cleave any substrate. Others very specifically target one other protein that's lying around. And what they do this because, of course, proteins are all somewhat generic. They all have the backbone, and then they all have the side chains. And the way that they make, for this case, for polymer specificity, they all still bind the main chain of the protein in the same way, but they differentiate between proteins by having pockets where a specific side chain is recognized, or a sequential set of side chains. And what you can see in this picture, this shows 4 different serine proteinases, which have very different pockets, and those pockets make them specific for, in this case with trypsin, arginine or lysine, or in this case, granzyme asparglue. And so you get specificity by defining these side chain pockets while retaining. And of course, lastly, in terms of paradigms of evolution, you have convergent evolution of enzymes in the sense that unrelated enzymes can converge over, they start off completely separately, but they can sometimes evolve to generate enzymes that perform the same function. So this is functional convergence. And sometimes they do it in the same way as each other. Even though the folds of the protein are very different, the actual active sites are very similar. And this is this shows some example with xylanases, where you've got actually different folds, but the active sites are conserved. So we wanted to understand more about these families and how they evolve. And so we looked at some of these big families using the sequences, the ligands, the promiscuity, the reaction mechanisms, etc. And Nick Furnham, who was a postdoc in the lab at the time, he analysed 276, a lot of superfamilies of enzymes. So each of these families is one, if you like, there was one original enzyme somewhere, and that enzyme evolved to do different functions. The question is, are those functions the same or are they different? Now, in the vast majority of cases, the functions are the same, but there are changes sometimes. And sometimes they change in the ligand that they bind, sometimes they change in the chemistry that they do. And what Nick looked at was these EC numbers that I described before, the different classes, and asked, when we see a change in function, do these two related enzymes perform, are they both hydrolases, or have some of them become isomerases or something else? And he created an EC exchange mechanism where one enzyme can evolve to do the same structure. And you can see most of these changes evolve. A hydrolase is still a hydrolase, it just works on a different substrate. Whereas others, the hydrolase occasionally becomes a transferase. So you have a difference in the enzyme. And this was done. And what we found was that if we looked over all these different families, we found that the vast majority of them just change the substrate that they bind. So there'll be some variation of substrate. Some of them, a small number, change the reaction chemistry that's performed. So over evolutionary time, you can see that you get many changes. And so this sort of paradigm of changing substrate in evolutionary terms is kind of easy. It's not a difficult thing to change your substrate. It's much more difficult to change the chemistry that you do. So we then went on and thought, well, okay, let's look at these active sites and see if we can classify them. And Yanis Riziotis, a student in the lab, developed a template library for all enzymes. So he looked at the catalytic residues that we had in the MCSA, and on average, there are 6 residues in each active site, although often it's 2 or 3, and defined a geometric catalytic template. And in fact, active sites could be broken down into 3 residue templates, so that's easy. So basically, he took the data from all the databases created from the MCSA and described this template library from all different functional atoms. And it's really just a section, the coordinates are defined. So it's a 3-dimensional representation of the active site. And we ended up here with about 3,000 templates and clustered them. So you can see here, these are all the different templates defined by in this case, it's a 3-residue template. The first residue is asphalt glue, because obviously they're similar. Same for the second residue. The third residue is a histidine. These aren't necessarily sequential in the sequence. In fact, most often they won't be sequential in the sequence. But then you can get larger templates like those down here, which obviously don't recur very often. But some of them, as I say, sometimes we find templates that hit different sorts of proteins, as here, where the active site, in this case, glue, hiss, 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 and it's a zinc binding motif. And this motif is found in several different, in this case, we've got the 4 different proteins that all bind zinc, and they have a similar template. Here we have the classic serine proteinase triad with the serine, the his, and the oxyanion hole that comes in again for unrelated enzymes. So in a way, this is going beyond. I've spent a lot of my research life looking at protein folds and general topology and architecture. This is now saying, well, actually, because we know these enzymes evolve new functions, we can't just look at the fold. We have to zoom in onto the active site and find the structure of the template here, and then see if we can find that template in the model. And this was work actually done some time ago by Roman Laskovsky. And what we found when we scanned the human genome, we found we had templates for enzymes, but also for the binding here. And we found about 14 examples in the human genome where the template hit and we could define a function for an enzyme which hadn't previously been recognized just by doing a fold analysis. Oops, sorry. So just to conclude this bit on enzymes, why we inevitably got drawn towards enzyme mechanisms. And this is something which I actually had avoided for many years because, as I said, I'm not a chemist. And so I wasn't sure that this was what I should be trying to do. But I still felt we need, in order to understand enzymes, we need to understand their mechanisms. And this is at the heart of green chemistry for anything. And if we want to design new enzymes, then we should understand how the ones that we know about actually perform the magic of converting a substrate into a product. And so we decided we would look at enzyme mechanisms. And part of, enzymes have many uses. Obviously, in our bodies, we wouldn't be here if we didn't have enzymes, but they have many commercial uses as well, which are shown in the slide. And so we decided to try to use a bioinformatics approach to study enzyme mechanisms. Now, if I think of enzyme mechanisms, I think of quantum mechanics and QMMM calculations and a lot of difficulties. But I thought, well, let's find out whether there are some, if you like, rules of catalysis for proteins, because proteins are they're spectacular, but they're also very limited compared with the chemistry, general chemistry. And so the logic was that, well, perhaps we can find some rules for how the steps of catalysis takes place. So the question was, if we get all this information about the enzymes, we have information about catalytic rules that we've derived from the database, we can employ a search algorithm, and then we can propose mechanisms for that particular enzyme. And this is what Antonio set out to do. For those who are technical, he developed it in Python, he used Django for the website, and he made heavy use of publicly available cheminformatics software. It's a whole field of cheminformatics. And we piggybacked on that and used that technology. And so if you think about, well, what is an enzyme mechanism? It's just the movements and interactions of the atoms in the active site. So this shows one such enzyme where we've got a reactant and a product. And we can see the different steps. So these enzymes, step 1, 2, 3, 4, show the movement of electrons with these curly arrow diagrams. So this is an age-old way of representing mechanisms in proteins. So this is a nice picture, but we wanted this captured in the computer, and that's what Antonio succeeded in doing. So for a given mechanism, he captured all the different steps that you can see down here, going from the reactant, where we start here, all the way along in 6 steps, in this case, to the product. And so this reaction was performed in these 6 steps. And so in this classic case for beta-lactamase, if you look at it, in fact, this is the reactant, that's the substrate, and this is the product. And there are 2 different hypotheses, well, in fact, more than 2, where you can get from the reactant to the product. You can go that way round, you may have a detour here, or you may go this way round to get to the product. And so there are different steps, and those we captured in the enzymes, and we use those, and this is shown in this paper that was published, now it's 2 years ago, gosh, it seems like yesterday, where we propose catalytic mechanisms. And from all these mechanisms, we can learn about catalysis. We can learn which are the most common transformations that occur in enzymes. And those are shown here. So as you might expect, proton transfer, including water, are the most common steps that we see in these 700 odd enzymes. There are also rules that involve cofactors, such as some of those shown here, and other common rules. So we can capture these and describe them. And then we can use the rules. So the way this works is you create your configuration, you pick one configuration, and then you apply the rules and say, do any of the rules that I've derived apply to this active site? If yes, then you generate possible intermediates. And so you go on. You then take the intermediate and say, well, what are the rules that apply to this? And you generate a whole set of different next step intermediates. And in fact, you do it from both directions. So you do it from the substrate and you do it from the product, and then you match them in the middle. So you iterate round, choosing possible steps. And this is a case, in fact, beta-lactamase, where you're trying to, which I showed before, and you're trying to find different potential steps. And this finds these different steps. And we tested whether we could predict the observed mechanism using this method. And in fact, it works very well if we use the rules. The problem is the coverage, that often some of these rules only occur in one specific enzyme. So it's not totally generic. Some of these reactions are generic. Some of them are confined to a single enzyme. And therefore, you can't, if you take that enzyme out, you can't find that mechanism in the data set. But nevertheless, we could predict some of them, but not all of them. But we have ways and means of improving that, which Antonio's working on. So in summary, this is a completely new way of thinking about enzyme mechanisms. We've created these rules. We've explored the chemical space around the reactants and products. There are many limitations, which you can read here, and I won't go through. But I think, obviously, we need to make the rules more generic. But it is a beginning, and Antonio's very strong idea is to combine this approach where we suggest possible mechanisms using a bioinformatics approach, and then you go and test these using QMMM calculations. So it's sort of part of the step. And of course, it would be lovely if we could just take an enzyme and predict what it did. It would also be lovely if we could say, I want this reaction, go and design an enzyme that will do it. And to do that, we need to be able to do this mechanism prediction. So I also wanted to broaden out, and I'm a bit conscious of time here, so I'll go through this rather quickly. Enzyme, I'm here at TIFR, fundamental research. I think this research on the enzymes is what I would call fundamental. It's trying to derive basic principles. However, we also know that with the new human genome and with all of our disease and health data, in terms of practicability, we want to be able to use the recent data to understand much more about human health, which is part and parcel of understanding life. And so I'll go very quickly through these. We obviously want to be able to interpret human variation. I mean, wouldn't it be fantastic if you could give somebody a genome and then you could draw a picture of this person just from their genome and say, well, what's good and what's bad about the genome and everything. I mean, it's scary in a way, and there's a lot of ethics involved, but it really would change the way we think about biology. It would be truly fundamentally changing. And so these variants, we want to know whether having the 3D structures can help in saying how we differ from each other, and what makes some people sensitive to drugs and others, and things like lifestyle choices. So we want to know about diseases, and we were involved in a study was called the DDD study, Deciphering Developmental Disorders. And it was run by the Sanger Institute across the whole of the UK. And the idea was to help to explain causes of diseases, which to me remains actually the goal of doing all this, which is basic research. Why does one person get a disease and another person doesn't? What happens in aging, given a given genome, etc.? So we looked at some of these mutations that were responsible. And this was just one protein. It's a beautiful symmetric protein, transducin beta-like 1X-linked receptor 1. And this protein came out. 4 children had variants in this protein that had developmental disorders. This was in a relatively small set. So this immediately implicated this protein. We looked at where the mutations occurred in this protein, and they all clustered together in 3 dimensions. And so that clustering gives you confidence that these mutations are the ones that cause the disease. And in fact, there's been a recent paper out doing exactly this on a broader way. We did it on several different proteins. We want to ask about aging. We know we're all getting up. I mean, even you guys, you young guys are getting older. You might not feel as if you are, but you will. And, you know, the population as a whole is much older. And of course, with getting older leads to a high increase in the rate of diseases, especially dementia, cardiovascular disease, and cancer. We know that. And so how do aging interact with diseases? Why is it when you get older that you get these different diseases? And I mean, the data, I still find this, I've had this slide for some years now, and I still find it amazing how strong the correlation is with age and certain diseases. And we don't understand why that should be. And so the scientific challenge in my mind is to understand the biological mechanisms of aging and then use that knowledge to intervene to improve late life health. All I'll say is that some years ago, we did this study where we tried to find drugs that might target aging. Because if you can stop somebody growing old, you'll also stop them getting these diseases. So it's sort of hand in hand. Now, whether this is possible, I think is still up for grabs. But my student, brilliant student, who's now Max Planck director, so a young director, they call them. We use gene expression data from human beings and from looking at expression drug perturbation expression profiles. We use that to identify genes that might affect aging. And we were very pleased to see that the ones in red in our list of which drugs affect aging are very well known drugs that affect that are known to affect aging in mice and flies. And we did some experiments in worms, actually. I'm not going to go into that, only to say that I noticed, I think last week, there was an exercise that showed the individual and additive effects of vitamin D, omega-3 and exercise on DNA methylation. And that, in turn, means that the biological aging of older adults seemed to be reduced. So these particular vitamins had an effect on how old your body was using the methylation clock. So that's quite complicated. So another aspect, I think, is we know that AI, I could have spent the whole time talking about AI and AlphaFold and everything, but we know this is going to be critically important. And the quality of the prediction from AlphaFold is really quite spectacular, in my opinion. And it really, I think, will help us to understand many different aspects of protein structure, because we now have 214 million structures alongside 2.5 billion sequences from metagenomes and microbiomes. So this again becomes a new world. We've got all these sequences, all these structures, and we can really use the structural data to help us understand it. So what's the future? So this is an old slide, but it does show that the cost per human genome is decreasing. Now this goes to $1,000 a genome. We now know it's down at $100 a genome. This is really not cheap still, but my goodness me, if we think how much we spend on our experiments, it's cheap. And so having this genome information means that there are many different aspects. Sorry, this was what I wanted to say. This is what we've got now. Now, almost every country, and I don't know what's happening in India, actually. I know somebody can perhaps tell me afterwards. There are big projects to determine human genomes for a selected set of individuals. That's happening worldwide. We have a huge opportunity to really link that information to all the health data, if we could do it. We have a huge opportunity, I think, with AI to interpret some of the complexities of that. So the opportunities in terms of this are enormous. There's opportunities for bioenergy, mapping biodiversity and barcoding life. Looking at the Darwin tree of life, you know, how many different species, this is in the UK, they're sequencing all the larger eukaryotes, the biotechnology. But to me, the real opportunity lies in the medical sciences for understanding disease and what causes it. We know that in many ways it is an ideal area. There's lots of data. We have many expert clinicians. It's a highly regulated profession. And the current approach is mainly based on previous experience. So this is a bit like homology modeling. You know, you've seen it before, so this is probably going to happen again. But that's a human estimation. To do it computationally, to me, is very attractive. But, and this is a big but, there are real challenges with the health data. We have electronic health records, we have hospital health records, we have cohort data, we have biobank data, we have Fitbit type data, we have social media data, but that data are rarely structured and rarely shared. And there is a huge challenge in building the data infrastructure in the UK for health data. There's the challenge of policy and ethics. Do you share this data? Because it's very, very personal. There's the challenge of data mining. What are the tools? How do we share this data which is scattered across the world? And obviously the regulations and security. And so there are major challenges in the data quality that are listed here. And the scale is just enormous. There's nothing the equivalent of CASP for predicting protein structures. Predicting protein structures, my goodness me, it's occupied me my whole life, but it's not complicated compared with health predictions. Much more difficult to do the health predictions. And the scale of the challenge here is enormous. And so in the UK, and I'm involved with this, they've set up something called Health Data Research UK. And the whole point of this is to make the data accessible for research. And this is, you know, a real challenge. It's in silos, it's scattered all around, there's the ethical process. And so in the UK, this is what we're doing. They're doing different things in different countries, and how this all comes together will be fascinating to watch over the next years. So I'm not got time, sorry. But I wanted to close by just saying, where's the future? Already single cell genomics has sort of overtaken the world. There's lots of single cell data. How we interpret it, still challenging, I think. Target validation for drug design, modeling metabolism, which relies on lots of different things will be important. Multi-scale modeling from, small to ecosystems, single cell proteomics, which is possible, but it's still developing. The novel application of all these molecular technologies from biodiversity to health and linking the genotype with the phenotype. So just to conclude then, I hope I've given you a bit of a view. I've scattered through a different thing and I could have made it very different, but I hope you've got a view of how using the computational approaches, you can really do fundamental research to explore how enzymes work, how the pathways work, etc. You can use the new AI technologies for then going further and looking at correlation, genotype, phenotype correlations. And really, I think I'm quite envious of you young guys. You've got the next 50 years to see how this is all going to pan out. I don't know how it's all going to work out, but I do think that using the computational approach will be at the heart of biology in the future and at the heart of medicine and the application of medicine in the future. And I think that's really important. So I'd like to thank my collaborators. These are people who've all worked on enzymes. You'll notice Neera here, who's been a long-term collaborator, who's Indian by origin, but she's been working at EMBL. Christine, my long-term Christina Rengo collaborator at UCL, and Samir, who runs the Protein Data Bank in Europe team and has been responsible for AFDB, the AlphaFold thing. And obviously EMBL and the Wellcome Trust for that, and my group. And lastly, I'd like to thank the family who funded my trip over here. One of the pleasures of being a scientist is the opportunity to visit labs, and I've had great pleasure yesterday at IIT Bombay, and today meeting various team leaders and students. And it's just so good to see the progress being made on all sorts of different areas and to try and fit it into one's mind about what's going on in the science of biology. So I have to say, I am delighted that I switched from physics to biology. This has clearly been for me, a great move, because I was really interested in how these things work. And I think now is the opportunity that biology can be modeled, can be quantified, can really be used in a predictive method. So with that, I will close. Thank you.
Thank you, Professor Thornton, for this illuminating talk. Now the talk is open for questions. Maybe we can take a couple of.
Thank you. That was a great talk. Your excitement is very contagious. Thank you. I just had a question about how much the model kind of takes into considerations of the cellular environment, because there are transient modulations to the conformation of these enzymes that is happening inside the cell, and the mechanisms may change. And you'd also mentioned that the functions will differ as per the active site. So how much is the model flexible into taking the microenvironment into consideration?
So there are ways of using the predicted AlphaFold models to look at different states, but you don't get the ultimate flexibility out. I've said this several times during my visit here already, but to me, the surprise has been how much we can say about protein functions from a single snapshot, usually, that you see in the crystal structure.
Yeah.
I think as we go forward, ensembles have been around for a long time. We've had NMR and people really haven't used them, partly because it's so complicated. And they don't take into account pH and, temperature and things like that. But it's like everything. You start off with a crude view of what's going on, and that will get refined with time. And I think what we will see is methods, and there are methods already, but they aren't generally used because it's so complicated. And the problem with the methods of predicting the impact of pH or temperature is validation. How do you prove whether you've got something right or wrong?
Yeah.
So I think in terms of function prediction and function analysis, we need better high throughput methods that will give us the data on which we can use some of the more sophisticated algorithmic approaches.
Yes, yes, thank you.
Thank you for this wonderful talk. So I have a few questions, both related to P450s. So as you mentioned, these class of enzymes, they share multiple substrates, and in plants we see so many of them. So do you think that this class of enzymes, they somehow compensate for the immunological inflexibility in plants, given that they don't have an adaptive immune system? And the second is that since they share so many different substrates, a single P450, do you think that the rules that you use to model would be more generic for them? And how do you account for convergent evolution? How do you model it in these classes?
Convergent evolution, I think I'll start at the end. To me, so one of the first discoveries was chymotrypsin and subtlysin. They're both serine proteinases. They are not related at all. Why nature chose to evolve two completely different enzymes to do the same job is not clear in my mind anyway. Maybe somebody else knows the answer to that. But I think at any one time, we know nature will evolve things that work. And so perhaps there wasn't a serine proteinase in the subtlysis, and so it developed its own. And those, because breaking up proteins is such an important part of biology, that this has been, tools that do that have been evolved several separate times. So you can have those completely separate events, maybe on different parts of the world or, you know, in the ocean and on land, I don't know. But I think there has to be some rationale for every time, because nature does not invent new things if it's already got something that it can change slightly in general. So that was the first question. With regard to the plants, I don't know a lot about plants. They're a world unto themselves in the sense that they have these different things. But I think it's, you know, it's very difficult to, they don't have the same immune system. They can't run away. Right. You know, this is a huge difference, if you like. They have to sit there in the soil. And I'm just intrigued with at home, of course, in the garden, we have daffodil bulbs. And they go through all of summer and all of winter. It's wet, it's soggy. The garden is squidgy because it's so wet. And these daffodil bulbs don't die. They can protect themselves against the fungi. And that's just one example. And all plants have the same problem. And so I think this, and it's just tinkering. You know, evolution is a great tinkerer. It tinkers on the outside. So the P450, the reason it can work, it basically just recognizes a hydroxyl group around it. And it's very promiscuous in terms of the rest of the context. So it works on anything with a hydroxyl, kind of give or take. And so that is a very useful thing for the plant to have, to enable it to survive in its context.
Thank you. Thank you.
Hi, Janet.
Yeah.
Yeah, thanks for the talk. I was interested in the prediction of enzyme mechanisms using bioinformatics. So I was wondering, do I see this as a simple network, or is there any energetics there which tells me what the rate determining steps are?
There isn't energetics there. And this, as we discussed before, energetics is the great leveler, because if you can compare energy. And Antonio, my colleague who's really responsible for Easy Mechanism, this search program, is absolutely adamant. I mean, he used to come into my office, you know, regularly and say, but Janet, we've not got any energy. We've got to have the energies. But we decided, because he was in Singapore and he worked for 2 years on one enzyme. setting the system up, doing the QMMM and trying to work through that. What this does is to provide the setup for looking at different possible mechanisms. And I think that is useful in that context. Whether it can go farther, because you can argue AlphaFold doesn't calculate any energies. And I've said before, you know, you've not got any metals there, and this thing still folds because it just learns. And so at the back of my mind, I'm thinking maybe if we had enough knowledge about these mechanisms, which must be, you know, the different things must be replicated in different enzymes, perhaps we could actually use it predictively. But then, of course, you'd want to do the QMMM. I think without a shadow of a doubt, you'd want to supplement it. But there's never one way to solve a problem.
Well, I was just wondering if you look at like the statistical weights, I mean, would you be...
So we haven't, and also we don't have that many. We have 745. Well, we have about 3,000 steps. And although that's a big number in my mind, it's still quite a small number if you're doing AI and statistics. But my hope was to use the rules of catalysis, which are only partial and not generic, and there's a lot more that needs to be done. But to use those rules to identify mechanisms that weren't right, because I'm quite sure in that database we have quite a few mechanisms that aren't correct. So, but we'll see how it evolves.
Thank you.
Ma'am, my question is that you explained that enzyme studies and drug repurposing is also used in anti-aging process. So that area can be expanded up to cancer treatment or treatment of various diseases if we study enzyme.
Sorry, I can't hear quite. Can somebody repeat it for me? Did you?
Ma'am, anti-aging.
Anti-aging and the drugs.
Drug repurposing.
Oh, cancer. So fundamentally, almost all biological processes, including cancer, of course, it's uncontrolled growth fundamentally, and that's often driven by enzymes. In fact, half of the drugs today are target enzymes. And so I think the idea of anti-aging, as I said, is that you know, there are various mechanisms for a person to say what their biological age is, apart from their normal, how many years they've been alive. And that has been used to say that with these drugs, that's what that paper was. With these drugs, these people looked as if they were younger than the ones who didn't take the drugs. They took the drugs for other purposes, not for aging. They were using them because of other things. And so I think just like the you know, these slimming drugs, which I don't know very much about, but they're all the rage. I don't know if they are, you know, they're really probably very important. But those are shown to have many different effects because they affect central metabolism, which underlies all the processes in the body. And so you are, you know, these processes, and this is the systems. You remember systems biology was the rage sort of 10 years ago. It still is, because we have to understand the system that we have within ourselves and then use it. And so I think enzymes are so fundamental to so many processes, and the drugs potentially modulate what those enzymes do. They rarely, it's interesting, if you look at mutations of catalytic residues, there are very, very few mutations of catalytic residues because you knock out an enzyme at your peril because you need it. Whereas many of the mutations that cause diseases are in second or third shell, where they modulate the activity. So they make the enzyme go faster or slower or whatever. And so, or grow, or they make the cell grow more. So this moving from, in one way, the big challenge is to move from the molecular to the cellular. And that's a jump that we can make, I think, now, because, partly because of the new imaging. I saw some beautiful imaging today of different cells. And to me, if we could link up what's going at the molecular level with what happens at the cellular level, that would be a huge step forward. So I don't know if that answers your question, but.
Okay, any more question for Professor Thornton?
Can I ask a question?
Yeah.
Yeah. So when you were describing the catalytic templates, I wanted to ask that what are the parameters used to characterize those catalytic templates, and how are they validated for suppose for a family of enzymes? And because there might be slight variations in the way the catalytic site is oriented for different enzymes, right, of a similar class.
So, essentially, it's based on the structure. And we take an enzyme in order to, what so the problem is, given an enzyme for which you know the sequence, you know the structure, and you know the reaction performed by that enzyme, can we predict the mechanism? That was what so this isn't ab initio, this isn't give me an enzyme, I don't know what it does, tell me what it does, predict the mechanism for how it does it, etc., which of course would be what you want to do. But at the moment, we can't do that. So this was a simpler thing. The data is taken completely from the papers, from the literature. So we've been building the MCSA since the early 90s, where we read the literature and it says these 3 residues are involved in catalysis, and there is preferably some QMMM calculations that show that it works, but that's not always the situation. So in the database, these mechanisms have a 1-star, a 2-star, or a 3-star. So we try to include what's known about the mechanisms. I often think mechanisms, they have an aspect, they aren't like structures where you can see them. There's an aspect of hypothesis in them, and it always worries me how accurate they are. But that was part of the reason for doing this analysis, to really validate the same way we validated structures, to try and validate these mechanisms. We aren't there yet, and I won't do this, but I hope some of my postdocs and students will carry on, and some really good chemists as well get in there and do some of this analysis. Okay.
Last question.
Yeah.
Good evening, ma'am. My question is, could you please shed some more light on the DDD studies that was done in UK? Like, what is basically the entire agenda with the mutations that have been happening with TB-LLR receptor?
Yeah, so the DDD study was carried out, and I think it might have finished by now. I'm not sure whether it's still going on, but what happened in, I don't know how much you know about developmental disorders, but normally young children present to hospital and they have some developmental problems. They're either not learning, or they're not walking, or there are problems. And in the past, only a few developmental diseases were known. And so what would happen would be that they would sequence the genes that were known. And they decided that the sensible thing to do was to look at the mutations. So they have trios. So they have mother, father, and child. If the mother and father are both healthy and the child is not, then it must be a mutation between, that's different from either the mother or the father. So you cut down the number of possible mutations. And then they would look through different genes would come out with mutations, were any of those genes already identified as being associated with a developmental problem. So that was the thing. They had, I don't know how many. When we did it, they had about 1,000, I think about 1,000 trios. So it's just a way, but it's a very obvious way to proceed, so that instead of just sequencing a bit of the genome, you do it all, and then you lose software and knowledge. So the actionable mutations in ClinVar, I don't know if you know ClinVar, but it has all the mutations in it, and some of them are known to be associated with diseases. So it's building up that knowledge as we go forward. There's a huge way still to go, and there are different ways of expediting that, there are just different ways of, depending on the disease and the background, the context of it all. So our role in that was just to see if the structural data could help us to, because you get, an average child will have at least 100 mutations. Which one's causing the developmental disease? That's the problem. And so and some of these mutations actually are actionable. You can give the child drugs that help them then develop properly. So that's the goal of this. So this is a worldwide project. This has to be a worldwide project, that we gather knowledge on our 20,000 genes, which mutations potentially can cause childhood developmental disorders. I mean, it's a no-brainer. And I think everybody would want that to happen. And I think it's ongoing in a broader way, not within the DDD. These things are terribly expensive to do.
Thank you, ma'am.
We thank Professor Thornton once again for talk and answers. So we end the program here, and tea and snacks is arranged in auditorium cafeteria. Please enjoy. Thank you.
Thank you. So after a shaky start, it worked okay. Thank you.